and not been shared.
No, they didn't isolate the virus. That's the issue.
What about live animal samples?
It doesn't tell you anything if they only test positive. I do not suspect it's coming from what we originally thought.
So we downloaded the sequences from the Chinese government, we put it into our proprietary algorithm that generates the vaccines, and in 2 hours we had the vaccine already designed and it went straight into manufacture.
So what has been your response here in this lab?
On the 10th of January, when we received the genetic sequence from the Chinese authority, we immediately designed a vaccine and immediately started manufacturing it and running tests on it in the laboratory. We're almost completed those now and the results are looking very promising.
How important was getting that genetic sequence from China?
Absolutely critical. It was key to what we're able to do here.
So I understand that from the genetic sequences to a model for the vaccine, you went through this in what, 3 hours?
So yeah, all we need is that genetic code. So it's just a series of A's and T's and C's and G's that make up the blueprint to the virus. We use a computer algorithm to generate the design of the vaccine. So we plugged in the viral sequence, and after 3 hours we had a fully designed vaccine on paper. And then after that, the stages to manufacture it kind of went straight into effect.
3 hours?
Yeah, absolutely.
Is that unusually fast?
Certainly, if you're thinking of traditional vaccines, they take much longer to design and to optimize and to manufacture. You're talking in the months to years type time.
Let me ask you what the Daily Mail is reporting. It says more evidence COVID was tinkered with in a lab. Now scientists find the virus contains a tiny chunk of DNA that matches sequence patented by Moderna 3 years before the pandemic began. Your reaction, Stefan, what can you tell us?
So my scientists are looking into those data to see how accurate they are or not. As I've said before, the hypothesis of an escape from a lab by an accident is possible. You know, human makes mistakes. So is it possible that the Wuhan lab in China was working on viruses enhancement or gene modification? And then there was an accident where somebody was infected in the lab and then infected their families and friends. It is possible. On the claim you just mentioned, the scientists are analyzing to know if it's real or not.
Yeah, I mean, I was struck by the line, it matched a genetic sequence patented by Moderna for cancer research purposes, Stefan.
Yeah, and that's the type of things that the team is looking at very carefully to know is it real or not. Okay. So it takes a bit of time to analyze all the genetic sequence.
And so we have to be grateful here that the Chinese posted the genetic code, because it was from that you were able to start working on it, understanding what was actually happening here.
Yep.
So once you have that code, why is that important? What does that unlock for you?
Yeah, absolutely. And I have to thank the Chinese authorities, because without that, we really couldn't have done anything. So because we use a DNA medicine vaccine, we need the genetic code to be able to design the vaccine. So as soon as we received that code, we were able to immediately start designing the vaccine. And in 3 hours, we had a design ready and raring to go.
3 hours?
3 hours to design a vaccine. And how have you managed to do that when in the past it took such a long time?
Yeah, so the vaccines that you and I are really familiar with, what we would call traditional vaccines, they're based on a protein, which is different from the type of vaccines that we develop at Inovio. And they take, in general, 2 to 3 years to develop. So you can imagine in an outbreak setting like what we're seeing in China, 2 or 3 years isn't going to help. Yeah. So that's where this new technology comes in, where we can really rapidly make a vaccine.
And so one of the most important things for you guys, I guess, is the investment behind it in order to manufacture something like this on that scale.
Absolutely. And we're really honoured to be one of 4 worldwide entities to have received a partnership funding from an organisation called CEPI, which is offices based here in London. And they were really, their mission was set up to respond to vaccine virus outbreaks like this.
Is this the one that has the backing of Bill Gates?
Exactly, yep, and the Wellcome Trust and a variety of different other government agencies.
A lot of work can be done, preclinical. One of the reasons we move so fast, we took 2 days to design the vaccine on the computer. Why? Because we had worked on MERS, another coronavirus, with Dr. Fauci's team for 2 years.
Yes. The data that we received are data that we got from what we call pseudovirus. So it's not the real virus. It is a virus that we have constructed in our labs, and it is identical with the Omicron virus. This is a very well-known study, and the first data that are coming are from this.
So this is Dr. Kate Broderick. Hello, Kate. She heads up research and development here at Inovio, and you can tell us about how you even start creating a vaccine. How does it work?
Absolutely, Tulip. So what you're looking at here is the actual DNA sequence of the virus from the outbreak in China. We received this virus sequence when the Chinese authorities put it online. We downloaded it. We started working on it immediately. And essentially overnight, we designed the vaccine that you can see here. The next step then is to put that into manufacture. So let's go and have a look at that now. Great.
But let's go to that second part of your question, which is a much more dark question. And that question is, what's really the agenda? Well, the fact of the matter is, and I've made this statement very, very frequently, you cannot manufacture a bioweapon and not say that you're trying to kill people. That's what a bioweapon is for. And the weaponization of the coronavirus, which officially started in 1999, when Anthony Fauci came up with this brilliant idea that he could come up with an infectious replication defective, and those are the actual words used, an infectious replication defective form of coronavirus. And he paid researchers at the University of North Carolina, Chapel Hill. He paid researchers there to invent a pathogen that did not exist. And that's the reason why I'm very careful with the language that I use. You'll notice I'm not saying a virus, and I'm not saying a vaccine for a good reason. We are not talking about a virus. We are talking about an engineered pathogen. We are not talking about a vaccine. We are talking about the introduction of a computer simulated code in the form of mRNA, but it's a computer simulated code not to stimulate your immune system, but to turn your body into a factory producing S1 spike proteins similar to those found in coronavirus. Slow that tape down and listen to it again. Exactly what I said. You're not injecting a piece of a virus. You're not injecting a viral fragment. You are injecting a code to make your body produce a pathogen. And then you're hoping, and that's all you're doing, you're hoping that the pathogen your body produces also elicits an immune response. But this is structurally so important for people to understand. The code that is injected into your body between January 12th and January 20th of 2020, that code was uploaded from a simulated server that was a simulation of the spike protein mRNA, and it was given to the Vaccine Research Council. And Kizzy Corbett, the doctor who made this stuff, at the very CDC, all the stuff submitted to the FDA, Kizzy Corbett said, and I'm quoting from her interview with Francis Collins, it's really cool because you don't even need much of a lab. You can build one of these on your computer at home. That's an admission of biological warfare.
Y Sanidad ha reconocido en un documento por escrito que no tienen el virus de la COVID-19 aislado ni cultivos del mismo. Así lo ha comunicado tras una petición al Ministerio de la Asociación Liberum y Biólogos por la Verdad.
El tema de las vacunas es más serio de lo que parece. Hemos podido saber, gracias a la Asociación Liberum Biólogos por la Verdad y un particular, que a través de una petición realizada al Ministerio de Sanidad ha reconocido en un documento por escrito, ya que estaba obligado por un juzgado, que
el Ministerio de Sanidad no dispone de cultivos SARS-CoV-2 para ensayos y no tiene un registro de los laboratorios con capacidad de cultivo y aislamiento para ensayos.
Nos explicaba en Dando Caña la bióloga Almudena Zaragoza la diferencia entre la secuenciación y el cultivo y el problema que supone no tener un virus aislado.
La secuenciación no es más que la recogida de material genético de los pacientes, se separa el ARN del resto de material que se recoge. Ese ARN entra en unas máquinas específicas que lo van, digamos, leyendo en base a una plantilla. El problema viene aquí, y es un problema muy grande. es que esa secuencia no se ha podido cultivar. Es decir, todos los pacientes positivos que tenemos en España no se ha confirmado la enfermedad con un cultivo viral y eso es importantísimo entenderlo.
Además de todo esto, el Ministerio de Sanidad descarga toda responsabilidad sobre diagnósticos y tratamientos en los profesionales sanitarios y reconoce que
los test por sí solos no suelen ser suficientes para determinar la enfermedad, requiriéndose una evaluación experta de la persona a la que se le ha realizado el test.
Así es. Esos test por los que se han tomado todas las decisiones de aislamientos, cierres de locales, aforos en iglesias, incluso confinamientos. En fin, medidas que han puesto en peligro los derechos y libertades de todos, como la presidenta de la asociación Liberum ha apuntado.
I gotta hit the beat. 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 I gotta hit the